Hello everyone, welcome back. I hope you are fine. Today we will talk about excitatory and inhibitory synapses. In the nervous system, both types of synapses are present, excitatory and inhibitory. So let's start. So first of all, I want to show you basic principles of synaptic transmission at the chemical synapse. In the nervous system, there are chemical synapses and there are some electrical synapses. In some another topic, some another lecture someday, I will talk about the comparison between chemical and electrical synapses. But today, let's talk about the chemical synapses. This is an example of a chemical synapse where there is a presynaptic terminal like shown here, and then there is a postsynaptic terminal. And at the postsynaptic terminal, there are some receptors present. Whenever action potential comes here, calcium entry results into the presynaptic terminal, and it causes the release of neurotransmitter which binds at the neurotransmitter receptors, and a response is produced. Like in this case, inward movement of the sodium is the response. Okay, so if you look at the synapses, their morphology and their structure, so synapses on the dendrites are often excitatory in nature. For example, these synapses which are present on the dendrites of a neuron is shown here. This is the cell body, these are the dendrites, and this is the axon. So such type of synapses which are present on the spine and on the shaft, they are excitatory in nature. And synapses on the cell body are usually inhibitory, like shown here. Exoaxonic synapses are those synapses which make connection on the presynaptic terminal, and such synapses, axon terminals, are often modulatory. So, like this is very brief about the morphology of the synapse. Further talking about the morphology of the synapse, they can be classified into two common morphological types based on and named after the E.G. Gray who described them. And these are Gray type 1 synapses, and these are excitatory in nature and use glutamate as a neurotransmitter, and Gray type 2 synapses. Type 2 synapses are inhibitory in nature and use GABA as a neurotransmitter. So the type 1 synapses are shown here, and compared to the type 2, you can see slightly difference in the structure that I will explain in the next slide. Type 2 synapse looks slightly different as compared to the type 1. And these are the exodendritic synapses, synapse present on the spine. So these are spine synapse and shaft synapse. Shaft synapses are usually less common, but these are quite common. We commonly see in the system. And we have already discussed that type 1 synapses are often glutamatergic and therefore excitatory in nature, whereas type 2 synapses, they are often GABAergic and use GABA as a neurotransmitter. Further looking at the structural morphology of the type 1 and type 2 synapses, so we can see that the type 1 synapses, they are excitatory and use glutamatergic as a neurotransmitter, and type 2 are inhibitory. They are often contacting the cell body like we have seen in the previous slide, and type 1 synapses typically contact specialized projections on the dendrites. These are known as spines and less common as the shaft dendrites. So now let's look at the uh, like structure and differences in between the type 1 and type 2 synapses. In the type 1 synapse, you can see round synaptic vesicles and flattened synaptic vesicles are visible in the GABAergic or inhibitory synapses. There is a dense basement membrane. There is modest basement membrane here. Presynaptic dense projections you can be seen as compared to the less obvious dense projection. And there is a wide synaptic cleft, narrow synaptic cleft, and large active zone present in case of glutamatergic synapses. And the postsynaptic density is quite complex, which is shown here. On the other hand, in the inhibitory synapses, there is a small active zone, and the postsynaptic density is thinner than the type 1 synapses. So this was all very brief about the synapses. See you again.